can try to explain again. So ligands are allowed to fit at all possible positions on the ligand binding site with all possible conformations. Many search algorithms are available to perform the task. So let me just try to explain this. Okay, let's say we have a molecule here. Okay, we just got a molecule here, small molecule, that we are going to see for how it can bind to an active site pocket. So here is our active site pocket, let's say. Okay, this region is our active site pocket, and if you are making this ligand flexible, they can have, same molecule can have different conformation. Okay, maybe like this. Okay, this is the same molecule, but this is in a different conformation. So the first conformer, let's say second, and so on, right? So what docking, the first step of docking does is it will take the first conformer and see where it can fit inside the active site pocket. Okay, we'll look for places where it can fit wherever you have defined the active site pocket, or if you are looking for the whole blind docking, it will go and search everywhere. Okay, so if you have done targeted docking, then it will look for where this molecule can fit at the active site pocket. So it will look for this place, here, 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 everywhere. Okay, right? The same thing it will continue with the second conformers. We'll look for at different position, right? So if you read again, ligands are allowed to fit at all possible positions, right? All possible position on the binding site with all possible conformation. So all possible conformation, we will go and look for the fitting inside the active site pocket. So many conformers with different poses will be generated. Okay, so several orientation of poses, right, are generated by the search algorithm inside the binding site. Okay, for each pose, a score is given by a scoring function. Okay, so some kind of score is given. So the first molecule, first pose for this first conformer, you'll be getting some score, right? Based on the score, the ligands are ranked and selected for further studies, right? So for the first conformer, there is some kind of score. Second conformer, there are also some score like this, and so on. So based on the score, you can rank this out. Okay, you will take those conformers which are having the highest score in the top list, right? 10, 8, 7, like that. This can be the first, or this can be the third conformer, this can be the seventh conformation, or something else, right? So you can list out those having the highest binding affinity or scores, right? So these are given by the scoring function. Okay, we won't go deep into this scoring function, but just to give you a overview, we'll see what scoring function is and how they calculate the scores. right? Two steps. What you have to remember is the first step, it give you poses, different poses at different regions of the binding site, and second, it will give score for those poses. So highest score, poses with the highest score you can list, you, you will be listed, and you can take the poses which is having the highest scores for further studies. So what are the search algorithms that are available? So we have this systematic search, so generally used for rigid body docking, and they search the conformations sequentially, takes long time and less practical for more flexible ligand. Okay, the stochastic method, in it is applied to flexible docking, uses random sampling and relatively faster. So some example of this algorithms are Monte Carlo, simulated annealing, genetic algorithms. So this two will be used, okay, in our case. Now the thing is, when you want to find this particular poses, okay, so you can do molecular dynamic simulation. So this is done for refinement of the molecular docking results, but you can also see which poses are stable. Okay, so you can run molecular dynamic simulation to find out which poses among the results from docking using molecular dynamic simulation. So what are the scoring function available? So we have this force field based, so it is ab initio based, so it will calculate from the very start, and it uses these non-bonded terms for classical molecular mechanics force fields, right? And you have this knowledge base. It uses the statistical observation of intermolecular contacts identified from structured databases. So it tries to construct, uh, uh, you know, uh, scoring function based on the experimental observation. Okay, so they take the uh, this experimental value or experimental parameters, okay, from the structured database. Okay, so this structured database they are mentioning are the experimental structured databases, so they are trying to include some kind of experimental data for calculating or getting this statistical potential. Okay, so another one is this empirical based scoring function. In this, they are trying to include, or they include the experimental data in the form of binding affinity. Okay, so they are using the experimentally determined binding affinity, to calibrate the molecular interaction. Okay, and from this they are trying to get the linear regression. Okay, some kind of mathematical model. Using this, they are calculating the score between the protein and ligand. Okay, so this is the equation for autodoc 4. Okay, so this gives you the van der Waal interaction terms. This gives you the molecule hydrogen bond interaction terms, and this is for electrostatic interaction term, and this is for desolvation effect. So one more term should be there. That is for rotatable bond. Okay, and this is for autodoc vena. So autodoc 4, what we are going to do autodoc 4 and also autodoc vena. You see the scoring functions, the equation for scoring functions are different. So the results from autodoc 4 as well as autodoc vena will be different. Okay, so in this autodoc vena they are using some kind of Gaussian function. Okay, so this is for attractive forces like the van der Waal interaction, and they uses this repulsion terms also, and then hydrophobic hydrogen bonding, and this rotatable bond. So W is weighted terms which are derived from the experimental binding affinity. Okay, don't go into this, that's okay. Just I'm telling you that these are the things that are there in talking. Okay, so when you say you know you have found a molecule, okay, let's say this is active site, you got a molecule that binds to the active site pocket, right? This is some kind of interaction. Let's say you have predicted to be having high binding affinity. Okay, so let's say -14 is your predicted binding affinity score, and when you did experimental study you also get, like, if it is in the terms